Hi guys, this is Kate McGregor, one of the 107 TAs. So today I'm going to be walking you through a bunch of plant hormone experiments that you would have done if we had in-person labs, but luckily we have an awesome staff here in a greenhouse, so we've done the experiments for you. So I'm going to show you kind of how we set up the different experiments, what the controls are, what the experimentals are, and then what they look like after. So we're going to start with auxin. If you guys you'll probably learn this from the plant hormone video that you'll also watch. But auxin is produced in the apical, in the shoot apical meristem, and it's basically responsible for the inhibition of lateral growth, so outward growth. So these two plants that have an apical meristem, as you can see, are extremely tall. They're just growing straight up rather than growing out, and that's because they have lots of auxin and they're doing great. Versus these plants, just to give you a side-by-side comparison, this does not have an apical meristem at all, so it can't produce auxin. So you can see it's kind of growing out and growing bushier, versus this one that's growing just super tall and skyrocketing. So this is a really nice side-by-side comparison. This has auxin and it has an apical meristem, and this does not have auxin or an apical meristem, so it's growing out. And that's one of the reasons, if you've ever gardened before and you want your plants to kind of grow bushier rather than up, you'll prune them. You'll basically cut off different pieces of the plant, and then you're basically cutting off the apical meristem so it will grow out rather than up. And so, just to look at that again, those are the ones without an apical meristem. They're very short and very bushy, versus the ones with an apical meristem are just super tall and they've grown and they're just going to keep growing up. So we actually have an experiment that we can do to show how auxin works. So basically what we have here is we're going to set up 3 different conditions. So right here we have our control plant. You can see it's labeled C. We're going to do absolutely nothing to that plant. So it's going to show us what would have happened in this experiment if we had done absolutely nothing. So we have something to compare our results to. Then we have this plant right here that we are going to give a lanolin treatment. Oops, and I just dropped the flag. And this is lanolin, and basically what this is, it's actually from sheep. But what it's going to do is we're going to basically cut off the apical meristem, which I will show you, and we're going to put this directly on that cut to kind of plug anything from happening there. And then we're also going to have an auxin treated plant, which will be this one right here. And what we have is we also have lanolin mixed with a synthetic auxin that's not as light sensitive as regular auxin. So it's going to be able to work. And we're going to do the same thing on that plant. We're going to cut off the apical meristem, which again, I will show you. We're going to swab a little bit of this on there. And this is going to act as a nice plug to keep the auxin right in place, right where we cut off the apical meristem. So let's do that. So basically, when you're looking for the apical meristem, you're going to want to follow the plant all the way up to the top. And on these plants, they have this nice kind of little opening, oh boy, right here, where you can kind of open that little pocket, and you see that little stem growing right there. That is the apical meristem. Again, that little pocket, we've opened it up, and this little bud right there, that's the apical meristem. And so what we're gonna do is we're gonna cut directly below it, right there. So now this plant no longer has an apical meristem. And same for this one. We're gonna kind of follow it all the way up to this little pocket right there. Again, you can open it up to find a little bud in the middle. Again, that's the apical meristem. It might be a little hard to see, but we're basically opening that nice little leaf pocket right there. And we're also gonna cut it off right below. And now that we've cut off both of the apical meristems on our auxin and lanolin treatments, again, doing nothing to the control, what we're gonna take the lanolin, which as you can see, if I can open it, is this nice kind of really sticky Vaseline-like substance. And I'm just gonna take a little Q-tip Gonna put just a tiny bit, oh that was a lot, okay, a little bit on the end of my Q-tip, and I'm just gonna come over to my lanolin plant and I'm gonna put just a tiny bit right on top. Basically just creating a plug that covers that whole surface where I just cut the apical meristem. So that one is good, that's been treated with lanolin. So now we're gonna treat the lanolin and auxin plant. So again, this one has lanolin to keep things consistent, but it also has a synthetic auxin. So again, same kind of lovely, sticky Vaseline-like substance. We're gonna take another Q-tip. We're gonna take some of this and we're gonna come over to this plant. And again, we're gonna just dab a good amount right on the top of it, creating a nice little plug, and also it's going to hold that synthetic auxin in place so we can see what that synthetic auxin might do over the course of this experiment. Okay guys, so we actually set up these experiments last week, so I'm showing you the result of a week's worth of just letting these plants grow normally after we treated them either no treatment at all with the control, we treated it with lanolin, and we treated it with lanolin plus auxin. So just before we go into any of the results, it's probably reasonable to assume that the control plant just grew normally, right? We didn't do anything to it, it was in normal conditions, it probably grew a little bit, but not too much. The lanolin condition, we effectively cut off the apical meristem and plugged it, so we really shouldn't see any growth there. Maybe a little bit of outward bushy growth, but we shouldn't see any taller growth. And then in our auxin treatment, auxin inhibits lateral growth and it promotes the plant to grow upward, so we might actually see that treatment have grown a little bit taller. So let's look at them. So obviously right here you can see from a little C flag, this plant right here is the control. You can see it pretty much didn't grow that much. It has a little bit of growth up here. But again, we didn't manipulate this plant at all, so it grew a little bit, which is just nice, showing us what the baseline growth would have been without any treatment. Then I'll kind of tilt this so we can see this. So this one right here, this plant is our lanolin treated plant. So you can see that it still has its nice little plug of lanolin right there. And you can see that it really didn't grow any taller. This is basically the exact same height as the plant a week ago. It grew a little bit out. You can see it started to grow out right here. Which makes sense because we basically cut off the apical dominant, so the only way it can really grow is out. So we do see a little bit of lateral growth, so that's cool. And then our tallest plant here is our auxin treated plant. You can see that last week it was about right here, and it's now grown all the way up here. So this plant, despite us cutting off the apical meristem, we gave it synthetic auxin, so it was still able to grow. And you can see it didn't really grow out that much either, and that's because we gave it auxin, so it was still able to grow upwards and suppress that lateral outward growth. So that's the results from this experiment. Alright guys, so the next thing that we're going to talk about are gibberellins, or gibberellic acid is the one that we're specifically going to focus on. Those are primarily responsible for stem elongation, and that's also something that you'll find in the wonderful plant hormone PowerPoint Cameron made for more information. But basically we're going to set up an experiment with 4 plants. We're going to have 2 conditions, and we're going to have a control and an experimental in each condition. So our first condition are these lovely mutant pea plants. They're mutant because they actually lack the ability to produce gibberellic acid. So that's just the reason why they're a mutant, it's just they cannot produce gibberellins. So you can see they're pretty short. They're kind of cute. There's really not much happening, and they really can't grow that tall because they don't have the hormone that allows their stems to become elongated. So we also are going to have a control that, again, we do absolutely nothing to. So we're just going to let this little guy sit for a week and come back and look at him. And then we're also gonna have a gibberellin treated plant. So we're actually gonna physically spray this with gibberellin, which I will show you. But first, we're gonna talk about our other condition, which is our wild type pea plants. So you can see this compared to the mutant, they're a lot taller. That's because these have fully functional gibberellic acid. They can grow as tall as they want, they're doing great. This is just a nice side-by-side comparison. This has no gibberellin, this has loads of gibberellin. So these wild type plants, we're also again gonna have a control that we do nothing to, we don't manipulate in any way. And we're also gonna have one that we spray with gibberellic acid. So I'll show you that now. So when you're spraying your plants with gibberellic acid, or basically anything, you wanna make sure they're separate from all the controls. You don't want any of that to go on the controls, because then it's not really a control anymore. So this is our gibberellic acid. So I'm just gonna come all the way back over here. Give it 3 or 4 nice squirts. Kind of let it dry out a little bit. And then I'm gonna bring it back and just make sure it really doesn't come into contact with the control. Same thing for the wild type. I'm gonna take it away from the other plants. Give it a couple nice squirts of gibberellic acid, and then we're gonna put it back. And then these will sit for a week, and then we'll come back and look at them. Okay guys, so we basically set up these experiments. Again, we set them up last week. So this is what a week's worth of growth looks like, and I'm gonna show you all of the different conditions separately. So first we're gonna start with our mutant controls. Again, our mutants have no gibberellic acid, and since it's a control, we did not put anything on them. So this is our before plant, and this is after a week. And you can see there's pretty much no difference here. They maybe grew a little bit just because they grow normally under normal conditions, but there's really not any stark differences here. They didn't really grow any taller, their stems didn't elongate, and that's because these are both mutants and they have no gibberellic acid. So they really can't grow any taller. So we don't see a difference, which is what we expect to see. However, let's look at our gibberellic treated mutant. So this is our before. Again, this is a mutant, so it doesn't produce gibberellic acid, and it's pretty short. There's not really anything happening. Now, this is our after. We sprayed this plant with gibberellic acid, and you can see that it really got taller. Its stem definitely elongated. So just the fact that we just sprayed gibberellic acid on this really made this plant grow quite tall and its stem really elongated. And if you think about it, it makes sense because this is a mutant, so normally it has no gibberellic acid at all. So we gave this gibberellic acid and it was like, oh my gosh, gibberellic acid, my stem is gonna elongate. And so that's why you can see this really stark difference in the mutant gibberellic acid treatment. So now we're gonna talk about our wild type plants. And again, our wild type plants have no mutations, they're just normal, they normally produce gibberellic acid. So let's look at our control first. So this was our before, this is our after a week. You can pretty much see that there's not really any difference. These maybe grew a little bit just as they were growing normally under the normal conditions, but we didn't do anything to these plants, so we really don't see any stark differences between our before and our after. They're wild type plants, so they normally produce gibberellin, so they have plenty, so these just grew normally. However, if we want to look at our gibberellic treated mutant, this is our before. Again, it's looking pretty good, pretty healthy, but this is our after. And you can see that it got a lot taller. Its stem definitely elongated from us giving it gibberellic acid. So you can see a nice difference between our before versus our after a week's growth. And since we didn't see any difference in the control, we can conclude that this growth was primarily because we gave it gibberellic acid, which is responsible for stem elongation. So now we're actually going to look at the effect of abscisic acid on some of these leaves from this lovely plant that the greenhouse gave us. So if you guys remember, abscisic acid functions in closing stomata. So those are those little pores on the underside of leaves. And so you can imagine if you close those pores, water can't escape, but also carbon dioxide can't get in. So there's kind of a trade-off between closing stomata to retain water versus having them open to have a little bit of gas exchange. But basically in this experiment, we're going to spray abscisic acid on our leaves to basically force them to close their stomata. So I'll show you guys the experimental setup, and then I'll show you the results from an experiment we set up last week. So we have this lovely plant right here, and you can see it has these really nice long, this part of the plant is called the petiole, and we needed a plant that had long petioles because we're going to be putting them in these green containers, and we need the petiole to reach the bottom of the container. So what we're going to do is we are going to pick our favorite leaf. I really like this one. I'm going to cut it off at the bottom, so now I have a nice leaf. And what I would do is I would put it in here. I would fill this up to this line with water, or I would fill it up with water and then mark where the water was filled up to. And then I would take tin foil and I would wrap this really tightly. And I'll show you what that looks like because I've already set these up for you guys. And then of course, we label everything in Bio 107, so you would also label which treatment there is. And there are 3 treatments in this case. So I'll show you what they look like because I've already set them up for you guys. So we have our control. And so this, again, this is what the experimental setup actually looks like. We have our leaf in here. We have it really, really tightly wrapped with tin foil. And that's because in this experiment, we're really looking at water loss. And so we don't want water to just be lost because we didn't wrap it tightly enough. So we're wrapping all of them really tightly to prevent water from being lost this way. Because the only way we want water to be lost is through the stomata on the underside of these leaves. So this is the control. This is basically just going to sit in this nice little rack, and we're gonna do nothing to it. We're not spraying anything on it, and we'll come look at it after a week to see where the water levels in this tube have gone. Then we have our abscisic acid treatment. So again, same thing, the leaf is in here, it's wrapped really, really tightly to prevent any water loss. It's labeled ABA for abscisic acid, and what I'm gonna do is I'm going to, again, away from all the other plants because we don't want to contaminate anything. This is our ABA. So I'm just gonna give it a couple nice sprays, kind of all over the leaf, kind of let it dry off a little bit. And then I'm gonna put it back in this rack. But what I'm gonna do is I'm gonna put it as far away in this rack from the control as possible. Because I don't want this leaf touching the control at all, because I don't want the abscisic acid in this leaf to go into the control, because then our control won't work, etc. So it's really important to keep things separate between controls and treatments in this experiment. And then finally, we have our humidity treatment. So again, same thing, this leaf is in here, it's wrapped really tightly, this was filled with water, we know where the water level is, I've labeled it humidity. This is actually gonna go into this humidity chamber because we kind of wanna see, so we have one treatment with abscisic acid, we have one treatment that is just the control that's gonna sit out in the same conditions as abscisic acid, but we wanna see what would happen if we put this in a humid environment. Maybe if there's lots of water in that humid environment, it would lose water slower than maybe the control. So this is just another way to see how plants lose water in a humid environment. So this is our humidity chamber. It's gonna be sealed. What we're gonna do is we are going to, well, we're gonna put that there. And then we're gonna take this off. We're gonna put our plant right there. We're gonna put this back on and press it down to kind of create a seal, and it's just gonna hang out in there for the whole week. So this is our humidity chamber condition, and then our control and our abscisic acid treatments are just gonna sit right there separated from each other for a week, and then we're gonna come back and look and see where the water levels have dropped to. All right, guys, so it's been a week, so I'm just gonna show you what we do to look at where the water levels are before we actually measure anything. So let's say this was our plant. What we would do is we would want to kind of peel away the foil layer to look at where our water level is, because if we take the leaf out, the leaf is already displacing some water, so that's why we're doing it with the leaf in. So we've kind of peeled back the foil and we're like, okay, that's where the water level is. And so we're gonna take our Sharpie and we're gonna mark it right there, and that's how we show where the water level has receded to after a week. And then after we've marked that, we can take off the tin foil, we can take off, and we can discard the leaf and just chuck that in there. Of course, we want to keep the sticker on so we know which treatment it is. But that's how you mark it. So I'm going to show you, and then also what we do now that we've marked the water level, we really don't need the water anymore. So we're just going to dump it. So I'll show you what our 3 treatments look like after a week. So we'll start with the humidity treatment. So I'm just going to show you guys this as a representation of what it looked like before. So they all had the same water level before, so this is the before for everything, just to give you kind of a way to compare it. So this is our before, and this is our after for the humidity treatment. So you can see if you put them right next to each other, we definitely saw a little bit of water loss. You can see this one definitely went down a little bit, but in the grand scheme of things, if we were to measure that, that would probably be maybe a centimeter, maybe 2. And that's probably because it was in a really humid environment. And so even if it was losing water, it could still, there were water molecules and like water vapors floating around in there. So it was just a humid environment. So there was plenty of water, so it didn't lose that much water. And then let's look at our control. So again, this is before, this is control after. So right away you can see that this water level has gone down pretty significantly. If you were to measure it, I would say that's a pretty good amount, and that's because this control wasn't treated with anything, and we just left these leaves open to the air. And so we didn't spray it with anything, we didn't give it abscisic acid to close its stomata, so its stomata were just functioning normally. And so maybe part of the time they were open and it was losing water, part of the time they were closed so it didn't lose a ton of water, but you can see it definitely lost water from before. And then we have our abscisic acid treatment. So this is pretty cool. You can basically see that these 2 lines are pretty much in the exact same place. And that's because we treated this plant with abscisic acid, and again, that forces plants to close their stomata. And so if the stomata are closed, the plant's wrapped really tightly with foil, and it's in one of these, there's really no other places for the plant to lose water. And so you can see that this plant did not lose any water, and so we can conclude that the abscisic acid really did cause those stomata to close. So it didn't really lose that much water. Yay! So now we're going to talk about another hormone, which is the hormone ethylene. So ethylene is actually a gas that's produced by a couple different fruits. It's produced by apples, which you see here. It can also be produced by bananas, which you see there, and it's produced by a couple different things. But basically what that gas does is it produces epinostic growth, which is an interesting term, but basically what it means is it causes the upper side, okay, so this is a leaf. It causes the upper side of the leaf to grow faster than the bottom side of the leaf. So you'll eventually get like this kind of curling or drooping downward effect if you have a lot of ethylene in an enclosed space. And that's what we've tried to set up here. So we have this like nice enclosed chamber, so whatever gas that apple is producing, so whatever ethylene it's producing, it's gonna get trapped in here, so its effects are really gonna be magnified on this plant. And same thing here, this does not have any ethylene in it. Obviously we haven't put any source of ethylene, but this is just to keep the experimental conditions the same between the 2 treatments. So it's a closed chamber here, and it's a closed chamber here with this apple producing ethylene. So it's kind of hard to see with the chambers on. So we're gonna break them open and see what the ethylene actually did to the plant. So I'm gonna take this off. And we're gonna take this one off. All right, so first what I want you guys to notice is, I think this leaf right here is a really great example of what ethylene's doing. You can see that the leaf is almost faced completely downward. And you can also see that on this plant, some of the petioles have drooped so far that they've fallen down. Things are drooping up here, things are drooping here, things are really drooping here. So this ethylene produced by this apple really caused epinastic growth here. And then versus over here, this plant looks pretty happy. It maybe has a little bit of curling downward, but that's normal for most plants. If you compare this curling down to this, you can see that this is extremely exaggerated, and that's because of the ethylene. This plant has a little droopiness, but this has extreme droopiness, and that's because it was exposed to ethylene gas. So this is the control. Not too many differences, still looks good. This is just a kind of sad, droopy looking plant.